Hello students, welcome to the class of Biochemistry. Myself Dr. Sureka, lecturer, Department of Biochemistry, SRM Dental College, Ramapuram. Today we're going to discuss about the monosaccharides, its structures and the properties. And this slide shows you the various sources of carbohydrates. The carbohydrates are very essential to derive energy to perform body's main functions. So today's lecture will see what is carbohydrates, especially the monosaccharides, its chemistry, structure and the properties. Coming to the definition of carbohydrates, the carbohydrates are nothing but hydrated carbons. They are also called saccharides. In general, the carbohydrates are referred as sugars. When you look into the chemistry of carbohydrate, they constitute of polyhydroxy alcohol groups attached either with the aldehyde functional group or a keto functional group. So here the picture showing the polyhydroxy alcohol group attached to the keto functional group. So there is the existence of other carbohydrates where the polyhydroxy alcohol groups attached to the aldehyde functional group. So either a aldehyde functional group or a keto functional group will be attached to the polyhydroxy alcohol. So when the keto functional group attached to the polyhydroxy alcohol, then the structure will be called as polyhydroxy ketone. When the aldehyde functional group attached to the polyhydroxy alcohol groups, then the structure will be called as polyhydroxy aldehyde. They are nothing but sugars. They are called as carbohydrates. Carbohydrates are present widely in plant and animal tissues. So it is composed of mainly carbon, hydrogen and oxygen. So in the picture you can see the presence of carbon, hydrogen, oxygen in both the glucose and the fructose structure. So when you come to the empirical formula of carbohydrate, which is nothing but the hydrated carbons, so here they are represented as CH2O. So here N also they have represented with, because each carbohydrate they differs in the number of carbon, hydrogen and oxygen, so it is represented as CH2ON. This slide shows you the functions of carbohydrates. The carbohydrates does various functions in our system. The one of the main functions of carbohydrate is to synthesize energy. So the good example for the carbohydrate which gives you energy is glucose. So glucose is a main component to derive energy, and also the glucose can be stored in our body in a form of glycogen, whereas in plant cells it is stored as starch. And also the carbohydrate, it serves as a structural component, example glycosaminoglycans. And also there is an existence of non-digestible carbohydrates. They are also called as dietary fibers. The example is cellulose, which is predominantly present in the plant cells. And also they serve as a component of nucleotides. The example ribose and deoxyribose. And also the carbohydrates involved in the detoxification reaction. The best example is glucuronic acid. Hence, the carbohydrates are classified into three categories. The first is the monosaccharide, which is a single sugar or a simple sugar. And second one is a disaccharide, which constitute of two sugars. They link via glycosidic bond. And third is the polysaccharide, where multiple units or many units of sugar they just link to form a polymer or polysaccharide. Here are the examples of monosaccharide, disaccharide and polysaccharide. So the example for monosaccharide is glucose, fructose, galactose, and for disaccharide it is maltose, sucrose and lactose, and for polysaccharide it is starch, glycogen and cellulose. In this lecture, we'll see in detail about the monosaccharides, its definition, classification with structures based on the aldose and the ketose unit, its biological importance, the projection formula, followed by the properties. Coming to the introduction of monosaccharide, the monosaccharides refers to one sugar. They are also called as simple sugars, which are highly soluble in water and sweet in taste. They are just composed of polyhydroxy alcohol units attached either with the aldehyde functional group or keto functional group. Since they are the simplest sugar, they cannot be hydrolyzed further, and they act as a building blocks for the synthesis of polymer or complex carbohydrates. The classification of monosaccharide is based on two characteristics. The first is based on the presence of functional group. The second is based on number of carbon atoms. Here shows the classification based on the functional group, where the monosaccharide is classified into two groups. The first is the aldose sugar, the next is the keto sugar. The aldose sugar found to have a aldehyde functional group in the structure, whereas the keto sugar found to have a keto functional group in the structure. This slide shows the well-known examples of aldose and keto sugar. Glucose is an example of aldose sugar. Fructose is an example of keto sugar. The aldose sugar, glucose, found to have an aldehyde functional group at one end, usually at C1 position, that is carbon 1 position. Same way the keto sugar, fructose, found to have a keto functional group, usually at C2 position. The classification is also based on the presence of number of carbon atoms. Depending upon the number of carbon atoms, they are classified as trioses, tetroses, pentoses, hexoses, heptoses. The trioses refers to 3 carbon sugar, tetroses refers to 4 carbon sugar, pentoses refers to 5 carbon sugar, hexoses refers to 6 carbon sugar, whereas heptosis refers to 7 carbon sugar. So the minimum number of carbons present in monosaccharide is 3. It starts with the trioses. This slide shows an example where glyceraldehyde serves as an example of aldotrioses, which found to have a 3 carbon in the structure with the aldehyde functional group. Ribose is an example of aldopentoses, which has 5 carbon in the structure with aldehyde functional group, and glucose is an example of aldohexose, which has 6 carbons in the structure and aldehyde functional group. And the example of keto triose is a dihydroxyacetone, which found to have 3 carbons in the structure and keto functional group. Ribulose is an example of keto pentose, which has 5 carbons in the structure and keto functional group, and fructose is an example of keto hexoses, which has 6 carbons in the structure and keto functional group. This table again shows few more examples of type of sugar with aldehyde functional group or keto functional group. If a sugar is having aldehyde functional group, then the sugar is called aldose sugar. If a sugar is having keto functional group in the structure, then the sugar is called keto sugar. So when you look into the names of the keto sugar, it has a word UL. Say for example, erythrulose, ribulose, xylulose, pseudoheptulose, and so on. So the most abundant monosaccharide which is present in our system is 6 carbon sugar, that is D-glucose. This slide shows the structural relationship between D Aldosis, which is nothing but the configuration of possible D Aldoses that arrives from the simplest monosaccharide, that is D-glyceraldehyde. So it is done by increasing the chain length via adding 1 carbon at a time. So what happens here? The aldotriose can give rise to 2 aldotetrose. 2 aldotetrose can give rise to 4 aldopentose. 4 aldopentose can give rise to 8 aldohexose. So this is done by just by adding 1 carbon at second position of the structure. This increases the chain length. Okay, when you look into the structure or configuration of the sugar, each sugar, they from the same type, they differs only at the second carbon. Again, this slide shows the structural relationship between D-ketosis, that means the keto sugar. So it starts with the simplest ketose sugar, that is dihydroxyacetone. Here, by inserting or adding one carbon at third carbon position, the length or the chain length of the sugar increases. So the keto triose can give rise to keto tetrose. Keto tetrose can give rise to keto pentose. Keto pentose can give rise to keto hexose. So each structure or configuration, they differs only at the third carbon position. Let us discuss the biological importance of monosaccharides. The monosaccharides have a significant role in our body. Here I have listed few of the important monosaccharides and its biochemical importance. Let us start with triose. The example of aldotriose is glyceraldehyde, which exists in the cell as glyceraldehyde 3-phosphate, which is an intermediate of glycolysis pathway. The example of ketotriose is dihydroxyacetone, which also exists in the cell as dihydroxyacetone phosphate, which is an intermediate of glycolysis pathway. The example of tetroses is erythrose, which also exists as erythrose 4-phosphate in the system, which is an intermediate of hexose monophosphate shunt, which is a carbohydrate metabolism. Pentoses example, ribose, deoxyribose, which is a component of RNA nucleotide and DNA nucleotides, which found in the structures of RNA and DNA respectively. Ribulose is formed as an intermediate of hexose monophosphate shunt, and xylose involved in the function of glycoprotein. Xylulose, which is also formed as an intermediate of uronic acid pathway, which gets excreted in the urine during the condition called pentosuria. Linkage is found in the heart muscle. This slide shows the biological importance of hexoses and heptosis. Glucose is an example of aldohexose, which is present in polysaccharide example starch, glycogen, cellulose, and also present in disaccharides such as maltose, lactose, sucrose. The glucose also found in fruits. The glucose is considered as a sugar fuel of life, which is all the living organisms, they depend on glucose to derive energy. The glucose gets excreted in urine during the condition called diabetes. Galactose, which is again an example of aldohexose, which is a constituent of milk sugar called lactose. The galactose gets converted into glucose during metabolism. The failure of conversion of galactose to glucose leads to an increased accumulation of galactose in the blood. This leads to a disease called galactosemia. Mannose, which is an example of aldohexose, which is found as a structural component of heteropolysaccharide. So the fructose present in fruits and honey, which is also exists as fructose 6-phosphate, and it is an intermediate of glycolysis pathway. Heptosis example pseudoheptulose, which is found as an pseudoheptulose 7-phosphate in the system, which is produced during the intermediates of hexose monophosphate shunt and the process of photosynthesis. The representation of structures of glucose and fructose are very important. Here the projection formula describes the structural representation of glucose and fructose. The structure of glucose and fructose can be represented in two ways. The first is the straight chain structure, otherwise called as Fischer projection. The second is the cyclic structure, otherwise called as ring structure, Harvard projection. We'll discuss one by one in the future slides. This slide shows the Fischer projection of glucose and fructose. Here in the structure of glucose and the fructose, the carbons are arranged one after the other as a straight chain. In this open chain structure, each carbon is named with different names. So the functional carbon is called anomeric carbon. The carbon at the end is called terminal carbon. The carbon before the terminal carbon is called penultimate carbon. These are all asymmetric carbons because here this carbon atom is attached to 4 different groups or atoms. So if the carbon is attached to 4 different groups or atoms, is referred to as asymmetric carbon. The other projection formula is Harvard projection, or otherwise called a cyclic ring structure. The Harvard projection are found to be very stable compared to open chain structure. The Harvard projection can be depicted as pyranose ring and furanose ring. Pyranose ring is a 6-membered ring, furanose ring is a 5-membered ring. So the open chain just undergo conformational change to form a ring structure. So here you have a 6-membered pyranose ring and 5-membered furanose ring. The crystalline sugar usually exists in open chain structure. When the sugar is dissolved in a solution, it just undergoes cyclization reaction to form a stable structure. Here, when a glucose is dissolved in a solution, the aldehyde group of the glucose, it forms a hemiacetal linkage with the 5th carbon, that is penultimate carbon. So here the first carbon, that is a functional group carbon, anomeric carbon, it just form a hemiacetal linkage with the 5th carbon or penultimate carbon of the glucose. So this undergo a conformational change to form a glucopyranose. The first carbon of the aldehyde group can also react with the 4th carbon to form glucofuranose, that is 5-membered ring. However, the glucopyranose is found to be more stable than glucofuranose. Similarly, the fructose powder exhibit open chain structure or a Fischer projection. When the fructose powder is dissolved in a solution, they undergo a conformational change to form a stable Harvard structure called fructofuranose. The second carbon, which is a functional group carbon, the keto functional group, involved in the formation of linkage with the 5th carbon in the structure. They form a hemi-ketal linkage. Hemi-ketal linkage is formed between the second carbon and the 5th carbon, and this undergoes some conformational changes to form a 5-membered fructofuranose. The second carbon of the keto group of fructose can also react with the 6th carbon to form a fructopyranose, which is a 6-membered ring. However, the fructofuranose, that is 5-membered ring, is found to be more stable when compared to fructopyranose. Here the picture showing the comparison of different representations of D-glucose. The first structure is the open chain projection or Fischer projection of D-glucose, where carbons are arranged one after the other in the linear form. The second structure is the closed chain projection, where the first carbon form a hemiacetal linkage with the 5th carbon. This structure is alpha-D-glucopyranose, which is a Harvard projection formula, where in the first carbon the OH is lying below the plane, which is represented as alpha. So this structure is the beta-D-glucopyranose, where the OH is lying above the plane in the first carbon. So compared to alpha glucopyranose and beta glucopyranose, the beta glucopyranose was found to be more stable than alpha D glucopyranose in the Harvard projection formula. Now we have moved on to the topic properties of monosaccharides. Monosaccharides has got physical as well as chemical properties. The physical properties are isomerism and Muta rotation. The chemical properties includes the action of strong acid, action of alkali, oxidation reduction reactions, and action of phenyl hydrazine. So let us discuss one by one. The first physical property of monosaccharides is isomerism. The compounds which possess same molecular formula but different structural formula are referred to as isomers. This phenomena is called isomerism. There are two types of isomerism, which includes structural isomerism and stereoisomerism. We'll discuss one by one. Structural isomerism have a same chemical formula but differ in their structures. Example, glucose and fructose. So the glucose and fructose have a same molecular formula, that is C6H12O6, but differ in their structural formula because the glucose contains aldehyde functional group and fructose contains keto functional group. So this type of isomerism is known as Ketosaltus Isomerism. The stereoisomerism is referred to as the compounds having same molecular formula but differ in the arrangement of atoms or its configuration. The asymmetric carbon in the structure of monosaccharide allow the formation of stereoisomerism. Here are the different types of stereoisomerism, which includes D and L isomerism, optical isomerism, epimerism and anomerism. Let us discuss the D and L isomerism. The D and L isomerism depends on the orientation of H and OH groups around the asymmetric carbon, which is adjacent to the terminal alcohol carbon. The carbon which is adjacent to the terminal alcohol carbon is also called penultimate carbon. In the penultimate carbon, when the OH is on the right, then the structure is a D series sugar. Same way, in the penultimate carbon, when the OH is on the left, then the structure is a L series sugar. So here we have an example, D-glucose and L-glucose, where in D-glucose the OH is seen on the right, whereas in L-glucose the OH is seen on the left side. So the D isomer and the L isomer of glucose are mirror images of each other. They are also called as enantiomers. Most of the monosaccharides in the living beings belong to D series. So the optical isomerism involves the optical activity of the sugar. So the asymmetric carbon enable the optical activity. So the optical activity is measure of the capacity of the substance to rotate the plane polarized light. So when the plane polarized light pass through the solution containing sugar, the sugar rotate the plane polarized light either to the left or to the right. When the light is rotated to right or clockwise direction, then the sugar is said to be dextrorotatory or plus. When the sugar rotates the light to the left or anti-clockwise direction, then the sugar is said to be levorotatory or minus. When equal amount of D and L isomers are present, the resulting mixture has no optical rotation or no optical activity. Then the mixture is referred to as racemic mixture or D or L mixture. Here are the pictures showing the optical isomerism or the optical activity of the sugar. When the light is passed through the sugar solution, the sugar can able to rotate the plane polarized right, either to right or left. So when the sugar rotates the plane polarized light to the right, then it is called dextrorotatory or represented with plus sign. When the sugar rotates the light to the left side, then it is called levorotatory. It is represented minus. Epimerism. The asymmetric carbon in the structure of sugar enable this property. The epimers are sugars which differ with one another only at the single carbon other than the anomeric carbon. So here is a picture showing the epimers of glucose, that is galactose and mannose. So when you look into the structure of galactose and glucose, they differ only at the fourth carbon. Similarly, when you look into the structure of glucose and mannose, they differ only at the second carbon. The other carbons seems to be same. So the galactose and mannose are epimers of glucose because they differ from glucose only at the fourth and the second carbon respectively. Anomerism. There are two forms of anomers available. alpha and beta anomers. As I already discussed, glucose predominantly exists as closed chain structure in a solution. So when you dissolve a glucose in a solution, the glucose undergoes cyclization reaction to form a stable ring. During the cyclization reaction, the anomeric carbon form a hemiacetal linkage with the fifth carbon. During this process, additional asymmetric center is created at carbon 1. So this is the asymmetric center which is being created during the cyclization process, which can give rise to two forms of isomers, alpha D glucopyranose and beta D glucopyranose. The structural differentiation between alpha D glucose and beta D glucose is the orientation of H and OH at the first carbon. In the solution, glucose will undergo a cyclization reaction to form either an alpha form of D-glucose or beta form of D-glucose. So the difference between alpha and beta D-glucose is the presence of OH group. If the OH is present below the plane at the first carbon of pyranose ring, then the structure is referred to as alpha D-glucose. If the OH is found above the plane of pyranose ring at the first carbon, then the structure is referred to as beta D glucose. This slide shows the closed chain Fischer projection and Haworth projections of glucose and fructose. Here the alpha D glucose and beta D glucose are mentioned, where in alpha D glucose you can see the OH group below the plane of the pyranose ring. Same way in the beta D glucopyranose, the OH is found above the plane of the pyranose ring. Similarly, the stable structure of fructose is fructofuranose. In fructofuranose alpha form, the OH is found below the plane. Similarly, in beta D fructofuranose, the OH is found above the plane of the furanose ring, which is a 5-membered ring. Let us discuss about the second physical property of monosaccharides, that is Muta rotation. The Muta rotation is the measure of change in the specific optical rotation which happens due to the interconversion of alpha and beta forms of D-glucose to form an equilibrium mixture. So when the sugar is dissolved in a solution, it either forms an alpha D-glucopyranose or beta D-glucopyranose. When the alpha D-glucopyranose is incubated for few hours, the alpha D-glucopyranose will interconvert itself into an beta form. Similarly, the beta D-glucopyranose, when incubated for few hours, the beta D-glucopyranose again interconvert itself to form an alpha form. So this happens through the open chain form of the sugar. This interconversion was detected by optical rotation. Usually, the non-reducing sugars cannot show Muta rotation due to the absence of free anomeric OH group. This slide depicts the Muta rotation of D-glucose. When a glucose is dissolved in a solution, it either forms alpha anomer or a beta anomer. The optical activity of pure solution of alpha D-glucose was found to be plus 12 degree, which means the alpha D-glucose can rotate the plane polarized light to 112 degree right. Similarly, the beta D-glucose can rotate the plane polarized light to 18.7 degree right. When the alpha D-glucose solution was incubated for few hours, it can interconvert itself into one beta form. The interconversion is possible just by opening up the chain, and again it undergoes cyclization to form a beta form. Similarly, when the beta D-glucose solution was incubated for few hours, the beta D-glucose open up its chain to again converts itself into an alpha D-glucose until it reaches the equilibrium mixture. When the optical activity of equilibrium mixture of alpha and beta D-glucose was measured, which was found to be plus 52.7 degree, which lies in between alpha D glucose optical activity and beta D glucose optical activity. When you estimate the amount of glucose present in the solution, the beta D glucose was found to be higher concentration, which is 63%, followed by alpha D glucose, which is 36%, and the open form of D glucose, that is 1%, which is a negligible amount. Now let us discuss about the chemical reactions of monosaccharides. The monosaccharides can react with chemicals to form different compounds. The monosaccharide can react with strong acid to form furfural. The monosaccharides can also react with alkali to form enol. The process is called enolization. The monosaccharide undergo oxidation reaction to form sugar acid, and also the monosaccharide undergo reduction reaction to form sugar alcohol. The monosaccharide were acted upon phenyl hydrazine to form ozone. First is the action of these strong acids. So when the sugar is treated with the concentrated mineral acids such as sulfuric acid or hydrochloric acid, the sugar loses water molecules to form furfural derivatives. These derivatives may condense with alpha naphthol of Molish reagent, orsinol of Biles reagent, isorcinol of Selinov's reagent to form colored complex. So this serves as a basis of the following tests such as Molish test, Selinov's test, Biles test. These are the best examples of furfural formation by monosaccharides. This slide shows the chemical reaction of Molish test, where when glucose is treated with concentrated sulfuric acid, the sulfuric acid removes the water molecules from the glucose to form hydroxymethyl furfural. So the reaction which is taking place here is the dehydration reaction. Again, this hydroxymethyl furfural condenses with the alpha naphthol of Molish reagent to form a colored complex. So in the test tube we can see a reddish violet color ring formation due to the condensation reaction with the hydroxymethyl furfural and the alpha naphthol. So second test is the action of alkali, that is otherwise called as enolization. So when the aldose and the keto sugars are treated with dilute alkali, it forms the enediols. Enediols are nothing but enol form of sugar which has two hydroxyl groups attached to the double bonded carbons. The enediols are found to be good reducing agents. So that reduces the cupric ions into cuprous oxide. So this is the reaction is the basis for Benedict's test and the Felding's test. So here is the picture showing the formation of brick red color from the blue solution. So first the blue solution turning into green yellow precipitate, then into orange precipitate, then into brick red. So when the solutions are showing brick red, which indicates a large amount of reducing sugar present in the sample. This slide shows the action of alkali on the reducing sugars such as glucose, mannose, fructose. They are the reducing monosaccharides. When these reducing monosaccharides acted upon by the dilute alkali, they form enediol. So the reaction taking place is the enolization reaction, where OH group is attached to the double bonded carbon. This enediol is a powerful reducing agent which reduces the cupric ions of copper sulfate present in the Benedict's reagent to form red cuprous oxide. So the tendency of red cuprous oxide is to precipitate out of the solution. That leads to a conversion of a blue color into a brick red color in the Benedict's test. So the reducing sugar give positive result to the Benedict's test and Felding's test. Monosaccharides undergo oxidation reaction to form sugar acid. So under proper condition, the aldohexose undergo oxidation reaction to form aldonic acid, saccharic acid and uronic acid. So we'll discuss one by one. Here is the formation of aldonic acid. So when the glucose is treated with a weak oxidizing agent such as a hydrobromous acid, which forms a gluconic acid. So the aldehyde carbon or functional group carbon will be converted into acid group, that is carboxyl group. So this structure is now called as gluconic acid. So the medical importance of gluconic acid is used as calcium gluconate, which is used as a source of calcium and injected intravenously during the calcium deficiency in order to raise the calcium level. This slide shows the formation of saccharic acid and uronic acid. The aldohexose, when treated upon by the strong oxidizing agents such as nitric acid, the first and the terminal carbon will be converted into carboxyl group, that is COOH group. So the resulting structure is now called as glucosaccharic acid. Similarly, for the formation of uronic acid, the glucose is treated enzymatically where the terminal carbon is converted into carboxyl group. The structure is called as glucuronic acid. The importance of glucuronic acid is, which is a component of heteropolysaccharide and a glycoprotein, and also the glucuronic acid involved in the detoxification reactions of benzoic acid, bilirubin, and also certain drugs. Monosaccharides can also undergo an reduction reaction to form sugar alcohol. Both the aldoses and ketoses gets reduced enzymatically or non-enzymatically to produce corresponding polyhydroxy alcohols. Usually the sugar alcohol produced as an intermediates in the pathways of carbohydrate metabolism. So in the next slide we'll show the formation of sugar alcohols from the monosaccharide. This slide shows the formation of sorbitol and mannitol. So the glucose and fructose undergo reduction reaction to form sorbitol. During the reduction reaction, the functional group carbons will get reduced to form an alcohol group. Now the resultant structure is called sorbitol. In diabetes patient, the increased glucose gets reduced to form increased sorbitol. The sorbitol has a tendency to get settled in the lens of the patient and leads to a condition called cataract. Similarly, the mannose undergo reduction reaction to form mannitol. The mannitol is used as an osmotic diuretic which enables the elimination of water molecules from the body to reduce the condition called cerebral edema. So the glucose and the fructose can get reduced to form sorbitol. Mannose can reduce to form mannitol. Galactose gets reduced to form dulcitol, and ribose gets reduced to form ribitol. So xylitol is a synthetically produced sugar alcohol which is used as an artificial sweetener in the chewing gum. The monosaccharides can react with the phenyl hydrazine to form ozone. All the reducing monosaccharide gives positive result to this test because the reducing monosaccharides will be having free functional group, that is aldehyde or keto functional group, which reacts with the phenyl hydrazine reagent to form ozone. The ozones are yellow or orange crystalline derivatives which are formed when treated with phenyl hydrazine. Each sugar will give a characteristic crystal structures which can be used for the identification and characterization of different sugars. Here is the formation of glucosazole from glucose. Glucose, upon treatment with phenyl hydrazine, it forms glucosazole. This reaction is a heat dependent process and involves three steps. First, it forms a glucose phenyl hydrazone, then it forms oxy sugar phenyl hydrazone, and later it forms glucosazole. So in the glucosazone, you can see the phenyl hydrazine which occupies the first and the second carbon. So the ozones are formed when the phenyl hydrazine blocks the first and the second carbon. Here are the ozone crystals of some sugars. The needle-shaped crystals, that is glucosazone, fructosazone, mannosazone, is formed for glucose, fructose, mannose respectively. The powder puff or tennis ball shaped crystal is formed for lactose, that is lactosazone. The sunflower petal shaped crystal, that is maltosazone, is formed for maltose. So the osazone formed from glucose, fructose, mannose are identical. Because when you look into the structure of glucose, mannose, fructose, the first two carbons only differ from each other. The lower four carbons are found to be same. As I already discussed, the phenylhydrazine found to occupy the first two carbons. So when the phenylhydrazine occupies the first two carbons, the structural dissimilarities between the three sugars will be disappeared. So the lower end of all the sugar seems to be the same. So all the sugar give rise to a needle shaped structure because of the identical lower four carbon atoms. The non-reducing sugar sucrose cannot form osazone due to the absence of free functional group. I hope this presentation would be useful. Thank you.